Hello, good day. I am Angieliquie Mejilla from BSN 1A. Um, Now let's talk about microbiology's chapter 7, microbial physiology and genetics. So first, what is physiology? Physiology is the study of vital life process of an organism. So what is the microbial physiology? Microbial physiology concerns about the vital life process of an organism, especially bacteria, because bacteria is suited for the study of the process or vital process of metabolic reactions of an organism. That's microbial physiology. So, um, let's move on to nutritional requirements. Um, Nutritional requirements of an organism. So, unlike us humans, microorganism or, or, or an organism needs essential nutrients for them to survive. That's what nutritional um, requirements means. So let's move on to categorizing microorganisms according to their energy and carbon sources. So first, we have phototroph. Phototroph uses light as their energy source. Chemotroph uses chemical as their energy source. Autotroph uses carbon dioxide as their carbon source. Heterotroph uses organic compound other than carbon dioxide as their carbon source. Photoautotroph uses light as their energy source and carbon dioxide as their carbon source. Chemoautotroph uses chemicals as their energy source and carbon dioxide as their carbon source. Chemoheterotrophs uses chemicals for an organic compound rather than carbon dioxide as their carbon source. So, that's categorizing microorganisms according to their energy and carbon source. So, let's move on to metabolic enzymes. So, metabolic enzymes, that's how we relate the apoenzymes, coenzymes, and holoenzymes. So, first, for metabolic enzyme, apoenzyme is apoenzyme can function as a holoenzyme when they are linked up in a cofactor, which is a non-protein cofactor or called as coenzyme. So, apoenzyme is a protein and the cofactor, which is the coenzyme, is a non-protein. When the cofactor is linked up with the apoenzyme, they will form a holoenzyme or a whole enzyme, a functional enzyme. So, apoenzyme plus the cofactor is equal a holoenzyme, a functional enzyme. So, let's move on to biologic catalyst. So, biologic catalyst, for example, endoenzyme and exoenzyme. So, endo meaning inside. So, endoenzyme is the reaction of an enzyme within the cell. So, exoenzyme, exo meaning outside. So, exoenzyme is the extracellular reaction outside the cell. So, let's move on to metabolism. So, metabolism, there are two types of metabolism. Anabolism and catabolism. catabolism and anabolism. So, catabolism, catabolism release energy. Anabolism requires energy. And then, there are the role of ATP molecules in metabolism. The role of ATP molecules in metabolism is that they are the major energy storing or energy carrying molecules within the cell. So, what is glycolysis? Glycolysis is the breakdown of glucose. There are factors that affect the efficiency of an enzyme. So, and the factors that affect the efficiency of an enzyme is, for example, when the pH is too high or too low, the enzyme will not function. So, enzyme di na makaka-functionhin maupay. Because of an itiya acidity is di rin na mimet. Di rin niya mamimet it iya maximum or peak level. Iya pag-react. Di rin makaka-reactin maupay because of factors that affect the efficiency of an enzyme. So, let's move on to bacterial genetics. So, bacteria, as we all know, all the bacteria may napogyap nira sarili nga genes. Ang itong sinisiri nga bacterial genetics. Nga tanan nga organism may nira sinisiri nga DNA, RNA. So, kung ang tao may da DNA, RNA, may genetic, and bacteria may dagihapon nira bacterial genetics. So we have the so-called lysogenic conversion. Lysogenic conversion is capable of producing one or new genetic product as a result of an infection of a temperate bacteriophage. So, we have the so-called transduction. Transduction, only small segments of DNA are transferred from cell by transduction. So, not totally, small segment like cell, it not a transduction. While transformation and is a whole or a bacterial genetic is transformed totally. So, ang transformation, ano sa ka-bacteria is nag-transformed genetically. Ang transduction, guti ay laan na babalhin. So, ang conjugation is a genetic material, usually in the form of plasmid, na nagtra-transfer an iya halophilus or sexphilus from the donor cell to the recipient cell. Amo pa to an conjugation. So, let's move on to mutations. There are types of mutations. Types of mutation is the beneficial, harmful, and silent. So, before we move on to this type of mutation, first, what is mutation? Mutation is an inheritable change of an organism. Regarding an iya DNA, nag-mutate. Amo pa itong mutation. Nag-change. Mutant is a phenotype in which is a phenotype nga nag-mutate. For example, it usa nga, for example, ako, mutant ako. Nag-change ako into something else. So, amo doon, mutant. Adto nga, pag-change ko into something else, ang process adto is mutation. Next is mutagen. So, mutagen is the one that causes the mutation to occur. So, for example, ako ang mutant, nag-change po ako. Pag-change ko, ang nakakos na akong pag-change is the mutagen. So, let's move on to the 3 types of mutation. So, what is beneficial mutation? And beneficial mutation is of benefit, may benefit. By the word itself, beneficial. And pag-mutate an organism is may benefit. Second is harmful mutation. Harmful mutation, harmful. So, ang pag-mutate, hanusa na organism, nag-cause hin harmful effect. So, may daya harm, nag-cause yan harm. Aniya pag-mutate. Well, the silent mutation, they have no effect on the cell. Again, beneficial, may the benefit, aniya pag-mutate. Unharmful, kumbaga, nag-produce siya in mutation han non-functional enzyme. And silent mutation, no effect at all. So, let's move on to genetic engineering. What is genetic engineering? So, ang genetic engineering, so, amo po ito ang genes, for example, ang genetic engineering, parehohan kan for example, may usa ka for example, Dr. Vicky Bello po, itiya anak, Scarlett is a product of genetic engineering. Ang ira genes, ang genes and mother and father, bagat inihapan mga chromosomes in order to produce good genes. Amo po itong genetic engineering. So, what is gene therapy? Ang gene therapy liwat is a process na, for example, may usa ka cell nga may sakit or infected na cell. Ito yan na infected na cell para maupay, gine-injectan nira, nag-i-inject po hirahin normal genes, nag-i-inject hirahin normal genes para makorek adto iya nga defected na cell or ito iya nga disease nga cell. Amo adtoan gene therapy. Gene therapy, amo itong pag-inject in normal gene ha defected na pag-inject in normal gene ngadto ha cell ngadtohan defected cell. Adto na process is called genetic engineering para maupay ang pag-produce in cell. So, amo po adtoan genetics. And then, so, that's the end of my discussions about Chapter 7, Microbial Physiology and Genetics. I hope you understand my explanation about this topic. So, thank you for watching. Thank you.